So the paper we're going to talk about today, very briefly, is a 2006 paper, highly diverse microbial structure in a remote central Tibetan geothermal spring does not display monotonic variation to thermal stress. This is, to me, a kind of a yin and yang paper. I think it's a really interesting question. And I think their experiments fail utterly to address that question. And so what they're proposing is that they're looking at hot springs somewhere completely opposite of where hot springs have usually been looked at. This is almost geographically opposite, only on the northern hemisphere, from Yellowstone National Park. And yet the springs are amazingly similar. So here's a picture of one of the springs there. I mean, this could be the Central Geyser Basin of Yellowstone. And so the one question they have is there such thing as geographic ranges for bacteria? When you look at a field guide for birds, one of the things that you always see is a map of North America and where the bird exists and where it doesn't. Are such things meaningful for bacteria? The answer is nobody knows. Some people would say yes. Some people would say that it's the environment that selects whatever organisms. The organisms are everywhere at some concentration, and the environment selects. There haven't been very many really good experiments that address that in any detail. The other question they're asking has to do with a standard ecological perspective about things, which is that an extreme environment is defined, the fact that it's extreme can be measured by the ecological complexity. Mild environments, like jungles, are very highly diverse, very complex populations with very detailed food webs, etc. Diversity is high. Extreme environments, like salt lakes, have very low diversity. In some situations, you have two species, Halobacterium and Artemia, the bronze shrimp, and that's it. That's the whole ecosystem. And so the notion is that harsh ecosystems have low diversity. And that in some way there's some gradation. I don't know of anybody in ecology that demands that there be a direct correlation between those two. I mean, certainly if you move north from here, you go through different kinds of ecosystems in bands. You don't see a smooth gradient from complex to simple when you get to the North Pole, right? You see one form of woodlands transforming to another in bands. And so I think what this is a straw man argument. Yeah.
So do you believe that there's a correlation between harsh environments and ecological diversity?
I actually think that's a bad question, because I think harsh is a judgment. I don't think harshness or mildness is a scientific measurement of anything. Extreme. Yeah. Well, what does that mean, right? I think that's a judgment. I think that the really important thing is that these environments, maybe selective pressure is primarily environmental rather than competitive, or something like that. So anyway, where's the picture of the spring? Here we go. So here's some outflows, little rivulets that come out of these springs, and they're taking various samples from the mats there and doing denaturing gradient gel electrophoresis. And so that's what these are. And so this one, which is a 52-degree spring, has one organism, as far as they can tell. This one at 63 has 3 bands, 66 has 1, 69 has 1, that may be the same thing, and so forth. Some of these communities are quite complex, some of them are quite simple. And then they go in with a razor blade and cut every one of these out in sequence. Yeah?
You can't look at 2 of those gels and say, okay, the bands in the same place and they're the same.
No, you cannot say that. any number of cases where two organisms have abandoned the same place. And so what they do is take every one of the sequences they get and try to get some kind of rough identification about what that organism might be. And again, they do this in the worst possible way. They do blast, and they find the closest hit. Now, in some cases, this is reasonable. I mean, if an organism is 93% similar to Pyrobaculum aerophyllum, you probably have something pretty closely. I mean, it basically is going to be phenotypically similar to Pyrobaculum. And so calling that a relative of Pyrobaculum aerophyllum is reasonable. If something is 73% similar to Thermoproteus, that tells you almost nothing. That could be anywhere in the archaeal tree. And so you gain very little information when you do that. And yet, they consider that to be like a Thermoproteus. The other thing is that they include in their blast search a series of uncultivated organisms. And so this one is similar to uncultivated Archaeon GA55, which gives you no information whatsoever because nothing's known about that organism, right? And they go so forth. Again, some of these are okay, 99%, but it's an uncultured Aeromonas. Okay, so it's an Aeromonas. They should have done something similar to what you guys are doing in lab with the classifier in the RDP, which would tell you, which would bin these into particular categories with some confidence levels. And then they do the proper thing, which is to tree out all these sequences and see where they come out. And they fall into the kind of classes you would expect: Crenarches, Uriarches. They get some thermophilic cyanobacteria, some chloroflexi. They even get some proteobacteria at the lower temperature parts of the spring, and so forth and so on. They really like their trees in this paper. Here's some specifics. The real take-home lesson, though, is these graphs. These are measurements of diversity. So they've looked at those denaturing gradient gels, and they're quantitating diversity based on the band, the relative band intensities, and they plot that versus temperature. The notion is that the temperature is the extreme of this extreme environment, and what they say they predicted was that that diversity would go down as the temperature went up. That was what they believe ecological theory would tell you. They do not see that, as you can tell. There is no correlation between temperature and diversity in this plot. Can you imagine what this might be? From my perspective, wouldn't you expect to see that drop best looking at the limit? 85 degrees is hot by our standards, but it is nowhere near the limit for how high temperature organisms can grow at. So I would like to have seen them do this same plot and take samples from the really, really hot parts of the spring. At this altitude, they can't go a lot higher than 85, but they could go to 90 or 92 degrees. That would be useful. The other thing is I would have liked them to characterize these mats as ecosystems in the same way that someone looking at a forest would say that this is a Midlands terrestrial deciduous forest, a maple-based forest, or where we categorize the ecosystems based on, in this case, probably the primary mat producers, right? This is a Chloroflexus rosiflexus mat, and so look at those mats as a unit. They don't do that. The last thing they do is they take their sequences and they blast GenBank, and they find that there are no sequences in GenBank identical to the sequences they pulled out. Their conclusion from that is that these organisms are unique, and therefore there is such a thing as geographic range in the microbial world. My argument would be you could go back to Octopus Spring today, fish out a bunch of sequences, and they would be unique, right? There's no comparison here behind us. And so I think they may be right, but they certainly don't prove it with this kind of data. Does that make sense? So again, this is the kind of paper that I find really interesting. You know, the questions are really, really important. But I think they do a poor job addressing those questions.